not a lot of interaction. Okay.
It's not an exact science. We have a separate color chart that's actually a better reference with an absolute 6,500 degree Kelvin white, but this tape serves its purpose. The chart would take up space in our forward storage, and it needs a T-arm, has to be held out there. We have a couple of them, but use the arm for expediency.
While they're still working on that camera white balance, let you know, question, is it this amount of marine snow normal for this part of the ocean at the depth? We are closer to shore, and we're also in a submarine canyon, and canyons are a place where you see a lot of sediment transport, which is a fancy way to say stuff coming down the channel of the canyon can kind of tumble down, even like avalanches. And being close to shore, the Astoria Canyon is an area of high productivity. When you have a lot of things in the shallow ocean, like phytoplankton or plankton, and then those die and sink, they come down to the seafloor. So we are expecting this to be a more deeply sedimented site rather than the carbonate ridge of our last dive.
Yeah, which is because of the canyon. And pouring everything out of the Columbia River and stirring it all up, all the turbidity, and also there's a lot of traffic out here. You know, boat traffic. It's quite a busy area. There are a lot of cargo ships that come in and out of the Columbia River, and this time of year, I think they're pulling a lot of grain out of eastern Oregon, eastern Washington. I think it's all going to China.
Yeah, all around the world. Yeah. Even, probably some down to the port of LA,
It could
just
be too.
Moving big,
a lot of freight.
Bulk.
A lot of bulk goes out of here. Yep.
I always like to say 90% of the things in your house have been on across the ocean voyage, even if you haven't been. There's even within the United States or certainly around the world.
Right.
The average is 90% of consumer goods.
Wow.
Have been on a freighter at some point, cargo ship. Chris, I have a question for you while we get set up here. Someone wants to know, does the marine snow and the debris in the water interfere with the sonar being able to find the bubble streams? Could you talk to us a little bit about that?
The marine snow and that sort of thing, and small fish and whatever, have very little effect on the sonar. The bubbles show up really well just because they're highly reflective. The water-air interface is super reflective, so that shows up much brighter than anything kind of watery floating in the water column.
We're all good with the stills cam.
Okay, roger. Okay.
Okay. Just setting everything up before we start looking around. Looks like we're not very, looks like we're almost on top of our target. We're going to be heading to
What's that for
waypoint.
position?
Well, freeze it again?
Yeah.
So you keep putting the shoulder you should keep everything down sort of like this.
Okay.
And then you have more range.
I wanted to put the elbow down,
yeah,
so
the shoulder needs to come up more.
Okay.
So to keep it keep it like that, and then unfreeze it.
And then lift it up.
Yeah.
Roger.
And pull it back towards you, right?
Okay.
We have a pilot in training. Kylie.
Yeah, unlike Alvin, we don't have official pit dives, training dives. So we just have to fit things in where we can.
So Bob can take a day off once in a while.
Well, we want a continuous stream of new pilots coming in, right?
Exactly.
As people move on, you have to have, it takes some time to get people trained up. So you gotta take advantage of the training periods when they avail themselves.
Is this okay, or should I modify it?
You're
that
good.
okay?
You're good. Yep.
Okay,
Yep.
blue light off.
You're planning on sticking around a few years, right, Bob?
Well, I'm not getting younger, though.
You and me both.
So Susan, what's the first thing we're going to do next?
We're going to try to find the seafloor marker, which was placed where we saw the hydrate in 2016 on our Nautilus dive. And that is waypoint 1. It's probably somewhere between waypoint 1 and 2. Waypoint 1 was our navigation spot in 2016. Waypoint 2, I believe, is where they saw the marker in 2019. So that just goes to show there's a little difference in navigation over the years, but it's a very small difference here.
Okay, Kylie, coming around at 258.
258, roger. I think Tim is the most popular person in the van.
He is, absolutely. Like,
he's a character.
Highly in demand, the data engineers. Yeah, if you are a longtime visitor and follower of Nautilus Live, you'll be familiar with the watch schedule and those team pages that roll over every 4 hours. You'll never see a few members of the team in that watch schedule because they are on call around the clock. And that is our expedition leader, our data engineer, our comms lead, and our deck chief, who just are available whenever you need them. Sometimes that means a lot, especially for the data engineer.
What?
Highly sedimented seafloor here. We're seeing lots and lots of sea stars.
Codfish. Sole, probably.
And we're heading on over to the waypoint. Looking for one of those seafloor markers. And looking at the video feed right now, you can tell why we put those seafloor markers out, because it's very hard to come back
on your map so you can
to the exact same place without them.
So keep your eyes peeled.
Okay,
What a seafloor marker will look like is a floating white disc.
I can't plot Latin lawn, but I can plot range and bearing, so.
Oh, I think, yeah, okay. Yeah, it's disappointing that didn't happen.
Yeah.
Seen several jellyfish. Sea stars, I remember we saw a lot of, oh God. What are they called?
Always grateful. Chris Ma from the Smithsonian has been tweeting along with us and IDing some of our hynoderms, some of our sea stars. Really an international expert on them. Super helpful.
Bridge now.
Oop.
10 meters. 230.
Oh, there's a little, wow, a big thorny head on the seafloor, which look quite similar to rockfish, but the thorny heads just sort of sit on the seafloor.
10 meters, 230.
I've been informed by fisheries folks that I know that we won't see any rockfish at this depth. They generally aren't seen in waters deeper than 500 meters.
There.
Oh, here we, up ahead of us.
Adding.
This is, you might want to slow down, guys. Wow, yeah, look at this.
Wow.
It's an amazing pit. A lot of times where these hydrates form, they can collapse. And so this is a huge collapse pit. We should put a marker down here.
Okay.
We're really close to the
It's
target.
probably, I mean,
Yeah,
the
actually.
marker might have been here.
Yeah.
And because when the marker was deployed, this was all flat seafloor. And I do not recall seeing a pit like this.
When they
So when the hydrate melts, it can collapse the seafloor around it. This is amazing. We're seeing a lot of bacterial mat and staining. You see the gray sediments. We see a lot of
Is that a net down
is
there?
that a net?
Yeah. Zoom in.
yeah,
Yeah,
let's
coming
zoom
in.
The kelp or a net?
Sargassum. Oh, there's no sargassum. It's a net.
Can you zoom in a little more? I wonder if that's hydrate right there. It would be hard to get at. It generally appears ice-like,
Yeah,
so.
that's just kelp.
Yeah.
Right?
Oh my lord. I wonder if this isn't where the marker was.
Could be off. We haven't really poked around the edges. It could be right next to it.
I don't.
We're really close to the target.
Yeah, and when we, like I said, when we deployed the marker, this was just flat. So it's very dynamic processes. Wow. Can we zoom in and see? I'm sure those are the
Zoom in on.
the clams that we're looking for. Oh my.
It's gonna
No.
be, tricky with all this stuff.
What are those? Those little white. I wonder what that is. Could you poke at that?
All right, zoom out.
That white spot right there? That might be exposed hydrate. Wow, that's amazing.
The last visit to this specific site was 2016. That's when we left the marker behind here.
Yes, and it was spotted in 2019.
Yeah.
And I don't, I'll have to ask tomorrow, but I don't hear any evidence that there was any kind of a collapse pit like this here in 2019. So sometimes when the hydrates melt, you know,
Is that like a big release all at once often, or?
I would think it would be. I mean, it looks like a single event.
I just had a
I
clicker
know.
on Hercules.
Oh, I hope it's not. I hope it's not garbage. Which
Can you zoom in,
unfortunately,
Ed?
Yeah.
which
Is
unfortunately,
anything happening on bubble just
Yeah,
now?
yes. We changed it though. We
Yeah.
changed the
That's a
camera.
different bubble cam. This is the other bubble cam, and it didn't have any
okay,
power glitches.
that is
It's like trash.
probably
Oh.
trash, huh? I don't know. Oh yeah, there we go. Trash.
Anthropogenic. Come out.
Disappointing.
Unfortunately, I think there have been very few dives that I've ever participated in where we haven't found some evidence of man on the seafloor. some bit of garbage. Yes, let's definitely you put a seafloor marker in here for collapse pit.
Yep, I already done.
And let's just explore around here a little bit more.
And you meant
And
a digital marker, right? Not one of the physical
well,
ones.
yeah, we don't have it. Yeah, we want to save that for sampling sites.
Right.
Yes, so I'll have to clarify, we have our physical markers that we're looking for, the triangles that are suspended on a chain, on a string with a chain link at the bottom so that it doesn't float away. And then we have our navigation markers. When we say put in a marker, they put in a latitude and longitude that indicates where we are if we find something of interest on the seafloor. Let's look over to the left here, or port, as we say.
This
Wow.
is fascinating.
Tamara, can you explain why?
Just a couple. Yeah, there's some slow bubbles coming up
Okay, hold on.
Yeah, I think so.
here and there.
Wow, it looks like craters
There's
of
a
the moon.
marker.
Yep.
Right here.
There it is. Wow, look at the growth or sedimentation on it.
Want to zoom in, Ed?
Yeah, looks like it.
273.
Concur.
Mm-hmm.
Message is authentic.
Lots of bryozoans.
Lots of fouling.
And our lead scientist has come in here and showed me pictures from 2019, and she said it did look like this in 2019. And she said that these pits can form from possible hydrate dissolution, some sort of possibly tectonic event, I don't know, that would cause these pits to form, but would upset the balance, the hydrate balance here. So that was where we saw the hydrate in 2016, so we need to poke around here and look. Let's look at the base of the marker first, and then
Okay, zoom in, Ed.
Coming in.
okay. Hey,
Hold.
come back a bit. Thank you. And that looks like hydrate right there. You see the white spots in the front of the camera here? That looks like hydrate. So our lead scientist, Tamara Baumberger's going to be coming in and joining us. Wow, it's so amazing how different the seafloor looks from the last time I was here, 5 years ago. This was just a flat seafloor. And now there are all these pits and lots of, you see bacterial mat, and the little tiny animals are probably, could you tell us?
Oh, what was that? Sorry, I was typing.
Just
Which tiny
the
animals?
little tiny shells.
The little shells are probably little snails.
Probably what?
Little gastropods.
Some kind of little gastropod, yeah.
Hmm.
All right.
Amanda Demopoulos writing in that they shook the debris off the marker in 2019, just kind of shook it a little, trash off. Look at the
You gonna talk, hon?
Ed, can you zoom out?
Yeah, let's just get a little, could we get some pics? I mean, I know that the still cam is running all the time, right?
Mm-hmm.
taking
Yeah.
continuous pictures, but while we're waiting for Tamara to set up, maybe we could get a better view, bring the camera up a little bit so we can see the marker on the pit and get some nice imagery. And we want to make sure we have the hydrate in the picture.
Allie, can you
Yeah.
bring that up on the view of the sexting cam up on something? I don't know what channel it's set to.
Taylor Ann, what channel is the sextant on?
So it has a still cam
I mean, I can't measure that.
button
Tamara.
back here for me.
It's right
Okay,
next
roger.
to it.
Got it.
Bob, when we came in here, did we see bubble streams right nearby?
No. Oh, you mean on the sonar? I wasn't looking for bubble stream.
Maybe we could kind of zoom around and look and see if we see any bubble streams very close by
Okay,
before
well,
we do any hydrate
That's what you
sampling.
want, right?
Did we get good shots of this? I wasn't looking at the sexting cam.
That's beautiful
Yeah.
right in there. Let's get some images like that.
So we're good on that?
Yeah, we are likely coming
Okay.
back and are going to sample it, but I would also like to know if there are some bubble streams anywhere close.
Okay.
It should be easy to find as it is
Switch
just
back,
next to the
switch
marker.
back.
So
I'm working on it. Oh,
Yeah.
wrong one.
And then reduce the sonar range down to like 10.
I think if we just kind of scan
10 meters,
around, it
roger.
should be very close by. I don't think we have to.
Range is 10 meters on the blueprint seeking.
Okay.
Back in
Okay, so
2016.
I'm going to come up a little bit.
Okay.
Coming up.
Coming up also.
Wow, beautiful shot. Okay, so now we're going to try to find the methane gas as opposed to the methane in ice form as a hydrate. Because Tamara would like to get samples of both in the same area so she can compare the solid phase and the gas phase of this methane.
Okay. 5 meters up. I don't see anything on here. You don't see anything on there.
Do you believe that's sampleable?
possibly,
We
yes.
keep getting
Yeah.
a blip.
Something right there too. Right?
Yeah. They're not moving either, right? Like,
So I'm holding steady here.
We saw a lot, just this morning they imaged, I believe, 2 large bubble plumes.
Watching Argus. Maybe you can kind of spin it around a little bit.
Sure.
Taylor, and where do I find the water temperature on any of these screens?
Where can you find the water temperature? Is that true? So are you logged into the science portal?
I am.
Yeah, you should be able to see it
Oh, got him.
there. Yeah.
Got him.
look at
Thank you.
this.
Oh, yeah. There you go.
Are you looking at Norbit, Chris?
No, it's on CAMAC 2.
Copy.
I'm having trouble pulling it up on the KV or on the video switcher. I've been just looking at it.
Oh, yeah. That's for sure.
Yeah,
What's the
Ed,
range on that?
we got a request.
15 meters.
14?
15, yeah.
We got a request for the lasers again, Ed.
Those are controlled by pilot.
Or Argus pilot?
I can do it. I can do
Oh, okay.
it. Cameras,
Hold on. Let me turn them on
Lights.
for
Oh,
you.
yeah.
lasers on.
Found it. You see it on there too?
Thank you.
Oh,
Oh, yeah, same thing.
okay.
Little
Yeah.
clapper on my
Yeah,
laser.
15 meters out.
Yeah.
So you want to go over there?
Yeah, sure. Have a look. And then
Okay.
go, if it's a good one for sampling, we go back to the hydrate and sample that first.
Okay.
Just need to get an idea of how the area is looking like and what kind of components we have here.
Okay, I'm going to match your heading.
Yep.
And you're swimming under me, basically.
All right, so what the front row is doing here is they're looking at their scanning sonars, a couple different ones on board that are looking out ahead of the vehicle. And because the difference in the density between the methane gas and the water is so stark, when an acoustic pulse, like the sonar pulse, goes out, you get a really hard return off of the bubbles. So we are looking with sound to kind of hear these things back, get that reflection back from the bubbles. And that helps us a lot because it helps us, those sonars can detect way further out than we can see clearly through this water, and to pick up small bubbles that are maybe tough
2
to
meters.
see.
We've got some matting on the bottom.
Drop the range down on that.
Drop the range. Raj.
Reviewers at home, I got my screen sorted here. We also have new live data, so you can check out some of these things like water temperature and depth by clicking the More Data tab
I'm coming down.
on the page.
Okay, Roger.
But it's 4 degrees Celsius, 39 degrees here on the seafloor.
45 degrees to port.
We're searching for the bubble streams. Not easy to find, but we're seeing all the signs. We see bacterial mat, shell fragments, a lot of gastropods, more of this pitted structure.
2 meters straight ahead.
Straight ahead.
Wow.
What is
that
pile
line?
of-
Yeah.
So
More. That's a different fishing line?
yeah,
Mm.
I don't think the other one was fishing line. Maybe this isn't fishing line either. Can we zoom in, Ed?
Yeah, coming in.
Here's the bubbles.
There they
That looks
are.
fine.
They're here, yeah.
Are there bubbles rising
Looks
out
like
of the
line to
fishing
me.
line?
Is that a line or is it natural?
Yes.
Yeah,
It's
it
a
looks
line.
like a line.
It's a line.
It's very fouled. Wow.
So Bob, do you think we could sample these bubbles or is it too close to the line?
Well,
Can do.
I'm not convinced that's line.
It's
I think going a little tighter on it helps.
Yeah, you zoom in more.
Where are they
So
coming
full
out of?
zoom.
Oh, right there.
It looks like a little fracture there, yeah.
I don't think that is line. I think it's some
It
sort
looks
of-
natural, right?
Yeah,
In a way,
yeah.
You think that's natural?
Yeah.
Oh, well, that's good.
Yeah.
What kind of a natural thing is that?
Rope.
Rope.
Not rope.
Rope covered with something natural.
Yes.
Oh, okay. Well, maybe it's a fallen kelp.
Sure looks like rope.
It doesn't, the way it's
Was
wound
old.
up, yeah, that doesn't look ropey.
Mm-hmm.
And you would know your rope.
Yeah, unfortunately.
Yeah. We've seen it before.
Help state, maybe.
Well, good. The reason I thought it was some kind of fishing line is that this is a heavily fished canyon.
Yeah, to the viewers asking questions about why don't we pick up trash? It's often floaty, and that's my scientific term for it, floaty. But that means it can escape from the boxes and take up bio boxes, contaminate samples. So it's
Foul or
There's some
thrusters
other bubbles here, Bob. I just saw more down in the foreground.
Where do you see them?
You want to zoom in more?
Yeah, let me just see, catch an eye on them again. I think they were down in this area, but it wouldn't be unusual for them to be underneath there.
Well, I don't think
You know,
it
under
would.
here, but
right?
I think I saw them in there. I'm looking again.
Right. Well,
Let me
zoom
do
in.
a push. You're sitting down, right?
Yep.
Of course, See them, they turn off.
That's all hydrate there, right?
Yep.
It does look like a chunk, yeah, but sometimes it's very difficult to see the difference between microbial mat and hydrate, but this looks like hydrate, I
There it
think.
is. So,
That's a big chunk of hydrate,
Yeah.
which is probably, I mean, a lot of it subsurface right beneath the sediment layer here. Obviously from all these pits that we're seeing, and
yeah, I don't see it now. This might be one of those where it burps every once in a while instead of a steady stream.
Yeah. Well, I don't see bubbles there
Yeah.
anymore either, right?
The other one stopped as well, right?
Yeah.
Let me go all the way in.
Oh, there
Oh,
you
I
go.
know.
there.
Okay, we just didn't see them. Okay. Yeah, this would be a good sampling site with having the bubbles and the hydrate,
All right. So we got those.
if it is hydrate.
You want to poke it?
Yeah.
let's poke it.
Full wide.
So for
Well,
the
if you-
hydrate sample, we need a fairly clean surface
Yeah.
of hydrate to
The
get
other
it in.
site
Yeah.
back there would probably have been better, right? Or you
It's
could,
probably easier for sampling, yes, but
yeah,
it's very tempting because we have the bubbles right here.
maybe you could scrape the surface off and,
I was gonna say, are you just looking for them as tightly paired as possible, or do you think kind of that 15-meter travel distance might be different pockets? What are your thoughts?
They could be different micro pockets, yes, but I mean, if I have hydrate and bubbles right next to each other, I prefer to sample like this, but
There it is.
it's not always the
You see
case.
it right
So
where the
basically
right
just
laser is, Bob?
as close as
Just a
possible.
couple bubbles came out of there.
can you use your hydrate sampler at a
No,
90-degree or,
we can try. The best is if we have it like straight over the sea floor, not like sideways.
Say, Megan, that's a great looking sticker.
Hmm?
That sticker looks great.
The new sticker, yes.
Yeah.
The 2021 Expedition Patch sticker? Yes.
Seahorse under the stars.
Yeah.
Yeah, it's sitting on the cover of the hydrate sampler. Yeah.
It does look great down there. Congratulations to our patch contest design winner, Alyssa. This was her design out of many submitted.
Yeah, thank you for everybody who submitted those. I got to see a lot of them. They look really great. And yeah, I'm happy with the one that we chose this year. It looks great. It's awesome.
Absolutely. And if you're inspired by this work to create art, we love to see it. We use the hashtag inspired by Nautilus Live. So if you're
Yeah, you want to zoom in, Ed?
drawing or painting along with us, creating things, share those with us. Feature them on social media.
Keeping you in frame.
so we are going to poke this and see whether or not it is hydrate.
It looks,
Matt.
yeah, bacterial mat sitting on reduced sediment. Yeah.
There's those bubbles again.
Yeah, over in the corner there.
Yeah. Adding.
Yeah, it's sometimes very hard to see from a
Yeah.
distance. Yeah.
Camouflage. Although there's some new exposed reduced sediment.
How about just back in there a little bit more, Tamara? You see the little plate that's sticking out closer to what I thought was...
Let me.
You can point on the.
Oh yeah, I can, can't I? Oh, this is awesome. I just love these new fancy things. How about that?
What are we looking at?
I don't know what it is. Yeah.
Let's see.
I think it might be.
You want
Some more plastic.
camera over? Yeah.
Into my reach here.
I mean, it's sticking out, so it's not like...
Need the pokey stick if we're gonna.
Roger.
Tamara, can you tell them what's so special about your hydrate sampler? Except that it's like the first in the world, like this.
They actually have other hydrate samplers, but this one, it's a gas-tight one, so we're sealing off the hydrate at the seafloor and then let it dissociate in a closed chamber while we're coming up through the water column. And this allows us to also measure the noble gases on the hydrate. And basically, noble gases are the ones you would lose first if you have some fractionation or.
Is that matte or is it trash?
I'd say trash, but might be wrong.
Yeah, it's like the other trash.
Like the other trash.
Plastic bag.
Yeah.
Some kind of plastic bag. Yeah.
And then right behind it is some of that braided natural material.
Oh dear. And what's that?
That doesn't act like rope.
Okay.
Like
okay, well that's positive.
Yeah, it might be kelp because I saw it looked like the air bladder of.
Yeah.
Not too sure.
Okay.
Just beyond reach.
It's all right. It's obviously not.
It's not hydrate.
It's not hydrate.
It's definitely not hydrate. Okay, what next?
Yeah. Let's look around in this corner a little bit more if there is actually some hydrate, and if we can't see anything, then we go back to our marker spot and try to sample this.
Like keep the pokey stick? Or?
Yeah, for now.
Oh,
Sounds good.
Keep that pokey stick handy.
Science.
You never know what you're going to have to poke.
Oh,
and now the bubbles are going, really going.
This is always the tricky part. Yeah. So in this case now, would like to have the bubbles and hydrates from the same site. So the thing is, if I now take the bubbles into our sampler, then we already have used up the bubbles. And if you are not able to actually sample the hydrate, then I have a full gas type, but no hydrate.
Reset?
So
yeah.
Yeah, no
Okay.
pair. Look at the bubbles coming up into our
yes, looks beautiful.
Wow.
All right, come on, hydrate. Let's
yeah, now I have
a matching pair.
Hard time to leave here.
Yeah, coming in.
Oh, yeah. Wow.
Oh. So I do have two samplers. So.
But we got two sides.
We got two sides. Hola.
That's nice. Obviously, you put down a nav target, Chris?
Yep.
Bubbles.
And it's also looking like they have the hydrate skin already, like how they're moving in the water column, these bubbles.
The hydrate coating?
Yes, yeah.
And is that just, you're looking at the slightly lighter color? Or how are you seeing that?
The way they kind of wobble. And then when it reaches the hydrate stability zone at 500 meters, that hydrate coating will disappear. And they'll turn into more like the normal bubble streams that we see.
So with the skin around, the reaction or the diffusion from the gas out of the bubble is inhibited as long as we have the hydrate skin around. So it allows us to move methane higher up in the water column until it gets out of the stability zone and the hydrate skin disappears, and then the bubbles start to exchange gas, like methane is diffusing out and oxygen and nitrogen can come in. Yeah.
This is the Wayback Machine, but in 2014, maybe 2013, we were working with the Geyser Group in the Gulf of Mexico, and they were looking at kind of oil-coated bubbles rising out and using extremely slow-mo cameras, and the bubbles kind of do this like wobble, you know, like a, I don't know, like they're flying saucer shaped and kind of teeter-totter back and forth. Is that the same thing these bubbles do if we were to look super slow-mo?
They would do something similar.
Okay.
Yeah.
Kind of like a raindrop. The way you draw a raindrop is like really not the shape the raindrop is. It kind of like flattens out. Those little wobbles.
We've also seen similar situations with CO2 coated bubbles in the Marianas,
Hmm.
and they just looked wobbly and sort of flat and stuck to the ROV arm. And
Interesting.
Yeah, I think let's leave those guys behind and hope for the best.
Okay.
Yep.
I mean, hope for the best.
And with that, I mean that they're still active when we come back. After we have sampled the hydrate.
After we find some spectacular hydrate
Yeah.
just on the other side of this little hill.
That's my plan.
Yeah.
I'm just going to stow this pokey stick.
Okay.
Like, it's easy to get to.
Okay.
Pokey stick has been stored.
Julianne, do you know what the little fish? Oh, not you. You're big, but the little all white ones with that same body shape.
The one like floating off the bottom right there, kind of next to the hydrate sampler, it's going to come out the other side. Or it's not.
Yeah.
That?
Yeah, just those little thin white ones.
I don't know if that's an eel pot or not. It looks too small.
Infernal pout.
Yeah, they look itty bitty. But yeah, there's some in the upper right.
Yeah, yeah, I see those.
Want to zoom in, Ed?
Yeah, coming in.
Those.
Oh, I'm just asking a question. You don't need to.
This guy?
Yeah, what is that?
Oh, no. I'm not too sure who that is. It could be an eel pot. I feel like they're a little bit longer though.
Yeah.
It's a juvenile.
Yeah, it's gone.
Okay, so what's the
Get my
plan
ID
here?
guide out.
Are we on the bubble hunt again?
More hydrate hunt. Yes, but if we could look at this whole feature here, like from the other side, and move a little bit around, if there's something that looks like hydrate, then we would pick it up from here, and otherwise we go back to the marker and try to pick up the one we have seen there.
So what will getting a hydrate sample, so the ice sample and the bubble sample from the same spot, what kind of questions will you be able to answer? What will you be looking at to learn from having kind of like a pair of samples?
On both of them, I will do the full chemistry with also including noble gases and isotopes. And then we are trying to find out if we can develop a fingerprinting method for the hydrate to find out if the gas that we are sampling is actually from dissociating hydrate, or if it was always in a free gas phase, like never incorporated in a hydrate. And for that we are using the ratios of noble gases. But some experiments have been done in a laboratory by other scientists, and it looks like the noble gases are actually fractionating when they are incorporated in hydrate. So we are trying to see if we actually see that in a natural sample, and if we can develop a fingerprinting method through that.
When you say fractionating, what do you mean?
That the heavy and the light isotopes get a different ratio, like that in this case the helium and the neon, or basically the noble gas fractionation, like some parts go away quicker than the others, like the lighter noble gases disappear faster than the other ones, and the heavier gets easier incorporated in things, like just the relative ratio between the noble gases, basically. That's what I mean with that.
So they don't kind of just freeze in place, identical. They're changing what portions of the gas end up locked in the hydrate.
Yes, yeah.
And would that also indicate whether or not it is thermogenic or biogenic?
No, that's a
whole
different thing. Yeah, there we would look at the carbon isotope composition. Yeah, but that with that we can't fingerprint if it was
It looks more
incorporated.
line-like, doesn't it?
Yeah, so
it looks
calling that line
looks
now.
more like line now.
I think it's kelp.
the great kelp line.
So this is a horrible angle because I'm just blowing into the side here, down the pit.
Yeah, we got the current behind.
Well, change it.
Oh, you want wanted to go around this side.
It was tomorrow. But I did too.
Definitely that, whatever that shredded plastic is.
There's a lot of it.
Yeah.
Okay, I'm gonna
And again,
hold here and wait till the cloud dissipates.
Or
We were,
wash.
yeah, we were.
Unfortunately, when you touch the seafloor with the ROV in highly sedimentary places like this is what happens. So we have to kind of wait for a little bit and let it float away.
Hopefully some hydrate will appear on the other side.
Voila.
Magic.
Yeah.
And the trash will disappear.
Yes.
Kylie will have found us something.
Again, we're not able to pick up that trash out here, but
Can you zoom in it?
one of the best things you could do if you, like us, hate to see this on the seafloor is just make sure trash is properly disposed of. You know, make sure trash cans have lids, things aren't blowing around and getting into streams. Streams go to rivers, go to the ocean.
That is line right
That
there.
is definitely, there's line in there, for sure.
I don't know what's at the
Tumble
bottom,
down
though.
canyons.
Mm-hmm. Yeah, and proper waste management is important to get it handled in places. Because once it's in the ocean, it's
Very difficult.
extremely difficult, borderline impossible to clean up. So,
Right.
But keeping it from getting there in the first place is a much more solvable problem that everyone can be part of the solution on.
And then it breaks down into microplastics. I read a statistic recently that said that there's so much plastic in our environment now that basically we're eating the equivalent of one credit card a week.
Taylor Ann, is that a stat you've heard? That's Taylor Ann's research.
Yeah, so last year I was actually out on the Nautilus collecting samples for microplastic pollution. And yeah, I will say that microplastics are ubiquitous within every marine system. I've read a lot of papers about that. It's even in the air around us. Sometimes you'll be seeing around like little fuzzies flying around. So that's why it's really important with my research that I try to prevent as much contamination as possible. So working in a room where there's not much air movement going around with a lot of microplastics possibly contaminating what I'm working with. And having a lot of blanks too, just in case if there is something that's possibly contaminating my samples that I know what that is.
Wow.
So I can kind of subtract that out of all the samples that I collected. So yeah, I've collected some water samples from the deep sea all the way up to the surface of the water column. Collected some sediment samples and some seafloor little grazers like fragile urchins, some sea pens that will feed in the water column to see if they're ingesting plastics, and if so, how many, what types, because we do know that they're here, sadly. And it comes from things like this. If this line is made out of like a plastic derivative, it'll just break down over time and create tons and tons and tons of microplastics. So yeah, every piece of plastic that's created will never fully be destroyed, just broken down into small little pieces that pretty much any organism could ingest or just kind of patch along with it, and something bigger can eat it. So it passes its way up the food chain to us, and then, yeah, we do end up eating it, sadly.
Just like they find them at the top of Mount Everest as well.
Yeah, they're all over the place.
Everywhere.
Plastic is pretty persistent.
And you're right, it never goes away. It just breaks down. Fish eat it. They think it's phytoplankton.
Yeah. So we should definitely invest in finding an alternative to plastic that's less harmful.
Yeah.
Okay,
Agreed.
so we're not seeing anything over on this side.
Yeah,
Want to poke around a little bit more?
we can just
Really look around.
look around or poke around, yeah.
Yeah, not poking, just look.
No pokey stick.
No poking,
Look,
no
don't
touching the
touch.
bottom. Poke around in a metaphorical sense.
Yes, yeah. Sorry, yeah.
So yeah, we have a lot of dirt that we collected on the bottom of the vehicle from setting down. So it falls off and we're not in a favorable position with the current. It comes into view and makes it hard to see.
Yeah, this all looks like bacterial mat here.
Like bacterial mat?
Bacterial mat, yeah.
Reduce the gray and is more reduced.
The sediment, yes, yeah,
Mm-hmm.
that's anoxic most likely.
Okay.
Okay, well I need to come around the other direction.
Sure.
To port.
Take a wrap out.
Thank you, Bob.
Yeah, so there's a lot
And
of
Kylie.
yeah, a lot of methane sitting in these sediments, in the sediment pore water dissolved in there.
Okay.
All right, so what's the plan?
Yeah, I think let's go back to the marker and try to sample that hydrate.
All right.
What direction is the marker at?
The marker should be maybe 190.
Bubbles right there.
Okay,
Some
we're seeing
bubbles.
more mat in front of us. Another, I think that's probably the same pit. That we looked at previously.
15 meters, 200.
Is it the
This
same?
is a different
Different pit.
Different
one. It's a
pit?
different pit.
Cool. Let's look at it. It is?
No, this is the same one.
Thank you.
Help
You
us,
said
Navigator Chris.
220.
Looks a lot alike.
Well, or yeah, 200 to the marker.
Oh, roger.
Okay.
Yeah, look at the bubble. Is that bubbles? I think it's
Are
bubbles.
they still coming out?
Yeah, look at the bubbles in Argus.
Yeah.
Yes.
That's fine.
Oh yeah, you can see them. They're still coming out.
Okay.
Just had to check really quick before we start sampling.
Yeah,
Yeah, that helped.
yeah, while we are moving to the back to the marker again, I can talk a little bit about the experiment USGS has been doing with colleagues not last year, 2 years ago actually, in 2019 on the Falcor, and that was also at this site. So there were 3 different type of equipment was deployed. One of them was a bubble box from Jens Greinert from Geomar in Germany. And this bubble box would have 2 cameras that are taking pictures of the rising bubbles, so to determine the shape and the size of these
So
bubbles,
like
and that's for calculating the volume and also quantify the gas that was released. So that was one part of the experiment. Then a second part was the gas quant that also came from Jens Greinert and Geomar, and that was a lander system with the multi-beam and CTD, and that was deployed in the same area, but about 50 meters away from
here, right?
Yeah, this pack.
About 50 meters away from where we had the bubble streams, and there the multi-beam would observe the bubble activity over time, and we had it down here for several days, so we would actually get some information of how variable the activity of these different bubble streams is, also if they're connected to tidal cycles or other variations. Then a third piece of equipment was a lander from the University of North Carolina. And in this lander system, we had the methane sensors that would measure methane concentration and also do incubation experiments at the same time to find out what the methane concentration over time is, but also the amount of the oxidation rate. And this was all three instruments were combined and deployed at the same spot. So that's a very interesting experiment that we were able to do during the 2019 cruise. And in addition, we collected some bubbles to get the gas concentration.
How long did those different experiments sit on the seafloor during that?
That was, oh, I'm not entirely sure anymore. I think it was maybe a week or 2 weeks, something like this. I would have to look up in the cruise summary.
But sort of like at the start of the cruise, you deployed them,
exactly.
and at the end you got them back?
The first few dives where we deployed everything and picked it up later in the cruise, yeah.
I love these jellyfish.
Jellyfish. They're so pretty. How about right in front of us, Tamara? Is that hydrate?
Not sure. So
Probably
far it looks like
reduced.
bacterial mats
Zoom
to
in.
me, but yeah.
Coming in.
To the right.
Holding.
Yeah, that's all fluffy mat.
Yeah, that's where I'm looking. Down. Yeah, that looks kind of similar to what we looked at before. You know, those little tiny things look almost like little tiny snails. That jellyfish wants to go. It keeps coming at us.
Hey, zoom out.
Okay, yeah, zoom back out. Thank you, Bob, or Kylie. Just interesting collapse pits all over the place.
That's where we were set down before.
Okay. We're heading back to the marker where we did see the hydrate.
Is it here?
Going to see whether or not it's sampleable.
No, when you say the marker, you mean the physical marker,
I
right?
mean the physical seafloor, the physical marker. I'm sorry. Yes.
Okay, that's the.
Yeah,
Yeah.
it was in this area here.
And you can tell looking at the bottom, that's where
we found it in this area. We might have been looking a different direction. That maybe that's not why we're not seeing it.
you can see the black spots. That is where Hercules touched down before. Not do that again.
Yeah, I can't remember if it was to the port side.
Zoom in.
Yeah, coming.
You might want to pull up a little bit. It was.
There's that
'cause there's the
plastic bag we
trash.
found earlier.
It would be higher. It wasn't in the pit. It was on the edge.
Wasn't it off to our port?
I think it was off to the port, yeah.
In front of that
From here.
next crater, hammock, whatever. Depression.
So yeah, pan to the port.
Should be coming in. Right there. See the line.
You can tell how hard it is to keep track of things on the seafloor.
Is that, there it is.
There it
That's
is.
coming in.
There we go. So now we're going to zoom on in and look at that hydrate, and Tamara's going to determine whether or not she can sample it. She wants to sample it.
Is it at the marker?
You know, I think it's the white spot coming into camera now.
Down
below.
slope a little bit?
This guy.
Right.
I love that.
Not quite sure, but
I think that you're right. Because we looked up by the marker earlier and that was